Identifying freshwater sponges along with creating a new database. Freshwater sponges are found all over the world and in the United States. There are non-moving vertebrates that are found in lentic, still water, and lithic moving water environments. They feed by filtering water through their bodies, and they're also environmental health indicators. The last Louisiana survey was conducted by Dr. Michael Poirier, 184 collection sites, 309 samples were collected, and 11 species were identified. And they were identified using the microscope. However, no DNA was collected. So the purpose of the study was to test and expand on the freshwater sponge using a reference database to identify Louisiana freshwater sponges. Materials and method. Since summer 2019, over 125 sites have been surveyed and 160 samples collected. Sponge reference database was constructed by Dr. Mary Miller from organisms identified by spicular morphology. To identify sponges using the database, we extracted the DNA. The COX-1 gene was used to amplify and sequence. Sequence were compared to reference strains using phylogenetic trees in MegaX. In the National Center for Biotechnology Information, known as NCBI, basic local alignment search tool, BLAST, was used to calculate the percentage similarity to reference strains and to further confirm the freshwater sponge species.
Results. During this project, our team was able to locate, identify, and confirm 4 new sponge species using the phylogenetic database MegaX. Table 1 represents species from both the 1969 study conducted by Dr. Poirier and the ongoing study conducted by Dr. Miller. Highlighted in yellow on Table 1 are the species our team confirmed and added to the phylogenetic database. Figure 3 is a phylogenetic tree. It is used as a tool to show the evolutionary relationship between different organisms. In this study, we used phylogenetic trees to help us identify organisms and their relationship to other sponge species. Highlighted in yellow are strains added to the database as a result of this study. Conclusion. Through this project, we now have increased knowledge of Louisiana freshwater sponges, 150 confirmed samples, and an expanded database with 4 new organisms. This research will facilitate future identification of freshwater sponge samples and other studies involving freshwater sponges. Further examination will be conducted with the winter sample collections to compare year-round data. Figure 4 shows the freshwater sponges that were collected and then identified using the phylogenetic database. Figure 5 shows pictures of sampling sites. Acknowledgements. We would like to thank the U.S. Department of Education Minority Science and Engineering Improvement Program and the Capital One STEM grant for funding. Matthew Hayer, Chao Vuong, and Charmaine Smith for Spicule Prep. And last but not least, we'd like to thank Dr. Miller, without whom none of this would have been possible. Thank you.